Muscle Metabolism 414, I'm Charla, and this is my handy dandy assistant. With the holidays coming up, I thought it would be a good time to talk about meat and meat alternatives. We all know how much Hans loves meat, milk, and all things animal protein, so I thought it would be a fun challenge to change his mind and maybe even put some tofurkey on his table this year. To start off, we know that all proteins have a varying amino acid content and bioavailability, or the proportion of amino acids in a protein source that our bodies can actually utilize. This chart here displays a wide comparison of essential amino acids and leucine content in common food. Leucine, highlighted at the bottom of the bars here, is an essential amino acid that has been proven to stimulate the mTOR pathway and promote muscle protein synthesis. You can see that animal protein sources are all higher in essential amino acids and leucine per serving. Taking the food out of food, we know that essential amino acid ingestion in their raw form with high leucine content have been proven to stimulate the mTOR pathway and induce muscle protein synthesis. Let's take it all the way back to week 2 when we reviewed the Fujita et al. 2007 paper and analyzed nutrient signaling in the regulation of human muscle protein synthesis. In this study, the researchers gave participants about 20 grams of essential amino acids with high percentage of leucine, about 35% of the solution, with about 27 grams of carbohydrates. In this portion of the results that I've demonstrated here, they saw the doubling of mTOR phosphorylation, high 4-EBP1, and S6K1 phosphorylation after ingestion, as well as lowered EEF2 phosphorylation. In the control group, none of these changes were seen. As a reminder of these pathways involved, I've included an image here from Hans' week 3 slides going over that pathway a little bit in the chunk that I wanted you to view. Overall, this study demonstrated that the primary regulator of mTOR signaling and muscle protein synthesis is amino acid availability. Soy, an example of a plant protein, still contains all 20 essential and non-essential amino acids, just with a lower proportion per serving. It has often been assumed that plant-based proteins are inherently inferior to animal-based proteins, given their lower essential amino acid content and lower digestibility. These claims are under-researched and not 100% accurate. While protein content per serving of tofu is lower in comparison to its chicken copart, eating more of it can make up for this difference. Additionally, when tofu is cooked, it has a higher digestible indispensable amino acid score, or DIA score, of around 95 to 99, meaning that it is considered a high-quality protein. Chicken's DIA score is only marginally higher, about 105 to 115, rendering both protein sources of high digestibility and quality proteins. Multiple studies, including one by Churchward, Venn et al. In 2019, have demonstrated that with soy, whey, and leucine-enriched soy protein isolates, all groups show equal stimulation of the mTOR pathway following resistance exercise. This type of study got me thinking, would these results be the same for each protein source if there was no mechanical stress on the muscles? Would these results be the same if they had utilized foods that are protein sources you might find in a supermarket? These questions pose a critical gap in our research. Though tofu is marketed as a meat substitute worldwide, it has little research to verify its efficacy on actually increasing protein synthesis rates the same way that common meats like chicken do. With this, it is imperative that we establish the efficacy of tofu on muscle protein synthesis and establish its anabolic properties. There are many environmental and nutritional benefits to consuming plant-based proteins, as I've shown here. It has been shown that eating plant-based proteins can improve overall human health. People consuming more plant-based proteins demonstrate reduced incidence of type 2 diabetes, obesity, cardiovascular disease, and even cancer. Eating plant-based proteins induces a lower impact on the environment, with less greenhouse gas emissions, offering a more sustainable option, indicating even more reason to partake in plant proteins. I personally have been trying to eat more plants-based since I took a medical physiology course and learned all about the long-term complications that meat can induce on overall health. However, I've been concerned about my protein intake, and I'm very curious if tofu can effectively stimulate my protein synthesis. Now I will get into the nutritional profiles of these two protein sources to explain why I picked them to compare in my study. Matched at 40 grams of protein means 125 grams of chicken and 232 grams of firm tofu. Tofu is much lower in cholesterol and higher in fiber, though it's a bit higher in fat. As for minerals, tofu packs a much more impressive nutrient content with lots of calcium, iron, magnesium, zinc, and copper. The essential amino acid content of chicken and tofu is decently comparable. Leucine levels are very similar at this protein content, being around 3.2 to 3.3 grams of leucine. With a larger amount of tofu consumed, it is obvious that tofu offers similar protein benefits with a nutritional kick. With this, I concluded that tofu and chicken should offer similar effects on muscle protein synthesis and prove interesting to study. Now into the nitty-gritty. As for the aims of my study, I'd like to assess the effects of 40 grams of protein from tofu, about 232 grams, on human postprandial protein synthesis rates. Number 2, I would like to compare the effects of 40 grams of protein from chicken breast, about 125 grams, versus firm tofu, about 232 grams, on human postprandial muscle protein synthesis rates. I've also reiterated the pathways here on the left to show the desired effects of nutrition-only study design, hoping to fully activate mTOR of both of these protein sources, as indicated here on the bottom, with nutrition alone. I hypothesize that muscle protein synthesis rates will be comparable between the groups due to the high bioavailability of both proteins and the substantial amino acid content at matched protein levels. As for study design, I modeled my methods off of a paper by Crow et al. That explored a very similar idea of analyzing the differences between chicken and a lab-created lysine-enriched meat substitute on postprandial fractional synthesis rates. First, subjects, n equals 15 for each group of chicken or tofu, will fast overnight and enter the clinic around 8 a.m. to begin testing. They will be screened for health, including height, weight, blood pressure, and body composition via our favorite, dual X-ray absorptiometry. Upon entry into the clinic, we will insert 2 catheters, one intravenously in the left arm for stable isotope amino acid infusion, and the other on the contralateral hand for arterialized blood sampling. We will take a blood baseline sample around 180 minutes before food ingestion. After this sample, we will prime the plasma phenylalanine pool and initiate a continuous intravenous infusion of phenylalanine. We will take 4 more blood samples at 90 minutes before food, 60, 30, and 0 minutes, or upon food intake. At 0 minutes, the first muscle biopsy will be taken from the vastus lateralis muscle of a randomized leg. Now, participants will ingest their tofu or chicken eaten with an 150 milliliter drink of water and phenylalanine to avoid dilution. Following the meal, blood samples will be taken at varying time intervals after 15 minutes and continuing every 30 minutes for 300 minutes. Postprandial muscle protein synthesis rates will be measured by taking biopsies at 120 minutes and 300 minutes, the end of our 5-hour experiment. Utilizing plasma from blood sampling and muscle tissue from biopsies, we should be able to calculate basal and postprandial FSRs and draw conclusions about the various protein sources and their efficacy to stimulate muscle protein synthesis. Some expected results are shown here in this slide, showing that FSRs between groups should be similar, very similar, indicating essentially equivalent effects on protein synthesis stimulation overall and broken up into the various time intervals. This slide here demonstrates similar results between various markers of mTOR between groups, further supporting the hypotheses that 40 grams of protein from chicken breast and firm tofu should demonstrate similar effects of protein synthesis. Overall, completion of this work will fill a critical gap in research on consumer-ready plant-based protein sources. This research will analyze the efficacy of plant-based proteins on muscle fractional synthesis rates and allow researchers for the first time ever to draw conclusions on whether plant-based proteins can, and maybe even should, be used as meat substitutes without any compromise on our protein metabolism. I know that I'll keep eating tofu with peace of mind now. I think my assistant would like to say something for all of us.
Bye, meat.
Just like they said. Alright, thank you all very much for listening to me drone on about tofu. I'm sure it's been very stimulating, and I hope that this presentation has opened your eyes to the fascinating world of vegetarianism. Have a great rest of your day and a lovely holiday this week.
We'll find it hard to sleep. Take one.
Eat tofu!